Hello everyone, so this is going to be a GCSE Combined Science Trilogy past paper walkthrough for Biology Paper 1 higher tier, and this was the paper that was sat in June 22. So I'm going to go through it and model it how I would have tackled it, and yeah, hopefully you can identify areas where you've got it nailed, but also areas that you need to go over and revise again. So question number 1 then. Bacteria can cause a variety of diseases in humans. What are two similarities between a bacterial cell and an animal cell? So you need to be thinking about what do they both have in common. Well, all cells both have a cell membrane. Yes, that is true. All cells would have a cell membrane. Both have a cell wall. That is not true. Whilst bacterial cells do have a cell wall around the outside, okay, they've got two layers. An animal cell, you should remember, just has a single outer membrane. Bacteria do not have a nucleus, so that one can't be right. They do both have cytoplasm. Those are two of the things that all types of cells have. Animal cells do not have plasmids, bacteria do because they don't have a nucleus. Okay, so that would be your 2 marks there. Salmonella food poisoning is caused by bacteria in food. Give one symptom of food poisoning, but you can't say vomiting or diarrhea. So therefore, it just leaves you with 2 other symptoms that you could say is running a fever or stomach cramps. Okay, just saying feeling unwell is not enough. Question 1.3. What was the name of the first antibiotic developed? Hopefully you should know it is penicillin, okay? Discovered in World War II that prevented many infection deaths. A child with severe bacterial infection was given a course of antibiotics. Figure 1 shows how the concentration of live bacteria in the child's body changed when taking the course of antibiotics. So you've got the time in days and you've got the concentration of the live bacteria in the body. You can see they started the antibiotics on day 0. The concentration of the live bacteria in the body continued to increase after starting the course of antibiotics. Suggest one reason why. So it's just a 1 mark suggest question. After I first start taking the antibiotics, why does the amount of bacteria actually continue to go up? Well, just got to put a logical or simple reason, so that is somewhat relevant. What is actually happening to the bacteria? Maybe at the very beginning, only a few bacteria are killed initially. So therefore it's not causing the number to go down. Or you could say there is simply a time delay, okay, before antibiotics reached or then actually killed bacteria. Okay, if you take a course of drugs, obviously you don't feel much better instantly. It takes a bit of time for the drugs to actually have their impact. Question 1.5 then. After 3 days of taking the antibiotic, the child felt better. There was still bacteria in the child's body. Why did the child feel better? Was it because the bacteria had become immune to the antibiotic? Well, if the bacteria had become immune to the antibiotic trying to kill them, then the child would actually feel worse. So it's not going to be that. The child had become resistant to the bacteria. Well, it would take our immune system to then become resistant to bacteria, and just because you're taking antibiotics, that won't make your body automatically immune to bacteria. It doesn't work that way. There would be fewer toxins in the body at day 0. So remember, if you've got less bacteria because they're then starting to die, the bacteria would be making less, there would be less toxins flowing about in your body. So it would be the bottom one. Suggest why doctors do not give antibiotics to patients with minor infections. Well, what do we not want to happen? If you're giving loads of antibiotics out to loads of patients, that would increase the chance that bacteria could become resistant to it. So it is to reduce bacteria becoming resistant to the antibiotics. So where it is possible, the doctors won't give out antibiotics if it's a minor infection and just let your immune system deal with it. Also for viral infections, antibiotics don't have an effect, so they shouldn't give it out for them either. Figure 2 shows a blood viewed through a microscope. So you've got your diagram here, and when I get something like this, I would try and recall what knowledge I know about it to begin with. If I'm looking at it, I've got loads of A, so that would be my red blood cell. B, the bigger type of cell, is my white blood cell. C is my plasma, the liquid that all the cells float in. And D are the small fragments of cells, which are your platelets. It then says a vaccine will stimulate the production of antibodies. Which part of the blood in figure 2 produces antibodies? We should know that it's our white blood cells that are a part of our immune system, so it should be B. Okay, white blood cells make antibodies and fight off pathogens. Which part of the blood in figure 2 starts the clotting process? Well, clotting process is happening if we get a cut and a scab will form, and that is the job of the platelets. So platelets do our clotting, and that would be D. Quick summary: red blood cells carry oxygen, white blood cells is our immune system, plasma carries other substances like CO2, glucose and hormones, and platelets clot blood. Okay, and those are your parts of blood. Question number 2 then. This question is about cell division. Write the biological structures in the box in the correct order of size. So we've got cell, chromosome, gene and nucleus. Well, hopefully we know that the biggest one would be the cell, and then the nucleus is inside the cell. What you then need to remember is that inside our nucleus, you've got your DNA, but your DNA is stored in structures called chromosomes. Now, chromosomes have short sections off them called a gene. So a smaller part of a chromosome is a gene, which would be the smallest, and then chromosomes are then inside our nucleus, and the nucleus is inside our cell. So figure 3 shows how a fertilised egg can produce specialised cells. So you've got a fertilised egg turning into an embryo, and then you've got process A happening and turning it into red blood cells, muscle cells and nerve cells. So I'll be thinking to myself, well, I've got a fertilised egg cell and it's just copying itself, so that would be mitosis. And then I've got an embryo turning into different types of cells. Now you might be thinking, oh, they're becoming specialised cells. The process is specialisation. That's what loads of people put in this exam, but that is not the correct answer. What you need to remember, an embryo is a stem cell. And stem cells are undifferentiated cells. Which basically means they're not specialised, they're undifferentiated, and they will become differentiated, and they then become differentiated through the process of differentiation. And that's the only word they would accept on this. Stem cells becoming differentiated through the process of differentiation. How many cell divisions are needed to form a 16-cell embryo from the original fertilised egg cell? Well, we know that when cells are dividing, they double up their number. Two daughter cells are made for every one cell. So you just double up 1, 2, 4, 8, 16. And then how many divisions has that taken? We've got 1 division here, 2, 3, 4. So it would be 4 divisions are needed in total to make 16 cells. 2.4. In humans, a fertilised egg cell contains 23 pairs of chromosomes. How many chromosomes will there be in each of the embryo cells? So you've been told it's a fertilised egg cell, and remember, fertilised is after the process of the sperm fusing with the egg. So all the DNA is together, and you've got 23 pairs. So how many chromosomes in total are there going to be? Well, it's 2 lots of 23, okay, giving you 46. Okay, 2.5 then. Figure 4 represents a cell cycle for a human embryonic cell. So you've got stage 1, stage 2, stage 3. Describe one change in the cell that occurs during each of the stages in the cell cycle. So this is just basically knowing, 1 mark for each stage, what happens at each part of the cell cycle. I'm just going to go through it and annotate it in this part here. Obviously, you'd write your answers on the lines. In stage 1, you need to know it's basically the preparation stage. In stage 1, the DNA is copied. Okay, so because the cell is going to split into 2 new daughter cells, you need another copy of the DNA. That's why it takes such a long time to copy it all. You could also say more mitochondria and ribosomes are needed or made. Okay. In stage number 2, when you've got your DNA copied, this is when your DNA lines up in the middle of the cell and is then pulled apart to each different side of the cell. So stage 2 mitosis, you can just describe it as DNA division. So the DNA then gets separated and half of it gets pulled to one side of the cell and half of it gets pulled to the other side of the cell. So stage 2 is DNA division. And then stage 3, this is when you've got your DNA copied and it's been separated. So what then happens is the cytoplasm and cell membrane separate. So cytoplasm and cell membrane split. Okay, and that's it. And that gives us 2 identical cells from what we then had at the beginning. It then says cell division is important for the growth of multicellular organisms. Figure 5 shows the mean height of boys and girls from birth to the age of 18. Okay, the question is, compare the growth of boys with the growth of girls using data from figure 5 in your answer. So we've got this graph here, and it is simply, we've got to compare boys with girls. So this is a classic question where you haven't got to know anything. This is a skill of describing clearly and comparing data from a graph. So I'm going to answer it in bullet points here. Obviously, you would write your answer on the lines in the real thing. But when you've got a graph like this and it's happening in time at the bottom, treat it like a story and try and include as many figures as you possibly can. So what I would be saying is, as my first point, is from birth up until halfway between 10 and 12, so at age 11, we can see that boys are generally, their mean height is slightly higher than girls. I can work out how much it is, so it's roughly 1 little square higher. So if I work out my scale, it's 20 over 5, which is 4. So I can say as my first point, between 0 and 11 years, boys are taller than girls on average by 4 centimetres. Okay, so I can clearly see for this whole length of time that boys are slightly above the girls. What I could then say, and that would probably get me 2 points there because I've said my point and I've supported it with data. What I can then say is where they then cross over. So at 11 years, boys and girls are on average the same height. and then I would say what exact figure that is, which is 140 centimetres. What I could then say is exploring the next part of the graph, and I can see now the girls' line is going up faster than the boys' line. So between 11 and 14 years, so between 11 to 14 years, the girls grow at a faster rate than boys. Okay, and then girls, so their rate of growth goes faster. They then cross over again at 14 years. At 14 years, so at 14 years, the average height of boys and girls is again the same at this time 160 centimetres. But then what happens is at the final part, we've then got our boys accelerating in growth, whereas the girls then plateau. Between 14 to 18 years, the height of boys, or height or growth of boys, accelerates and plateaus at 180, or maybe a bit lower, about 179 centimetres. Whereas girls' rate or girls' growth rate plateaus at 162 centimetres. Okay, so what I've done, I've broken this graph down into different stages and I've supported each stage with data. Now there are loads of different ways that you can break down this graph, but personally I would always just treat it as a bit of a story and describing it as time goes on. Okay, question number 3 then. Oh, did I miss a question? Sorry, I did miss a question. 2.7. Give one way that cell division by mitosis is important in fully grown animals. So in fully grown animals, we should know that mitosis, what do we need it for, is growth and repair of tissues. But because we are fully grown, we can't then say growth. So therefore, the only use of mitosis, when you're an adult, what does your body use mitosis for? It's just simply the repair of tissues, or replacing tissues when they die. Okay, and that would be the answer to 2.7. Question number 3 then. Amylase is an enzyme that digests starch. Okay, which organs in the human body produce amylase? So hopefully we remember the 2 places that all enzymes are made, which is the pancreas and small intestine. These 2 areas make all of the types of enzyme all of the time. Okay, so I would immediately be looking for these ones. So small intestine, okay, so it has to have these 2. So pancreas, small intestine, pancreas. So actually, just through process of elimination here, it's got to be the 3rd one, because these are the only 2, only option that has the pancreas and small intestine in. The 3rd place, which is given in the answer, is also the salivary glands. So anytime you're looking for where enzymes are made, you're thinking pancreas and small intestine. The extra place you make amylase is the salivary glands. the extra place you make protease is the stomach. Okay, so just make a little note to yourself. Protease can also be made in the stomach. Lipases are only ever made in the pancreas and small intestine. So the student investigated the effect of pH on the activity of enzyme amylase. This is the method they used. They prepared amylase solution at pH 5. They mixed the amylase solution with starch in a boiling tube. They dropped or removed a drop of the amylase starch mixture every 30 seconds and test it for the presence of starch. They record the time when all the starch has been digested. They then repeat steps 1 to 4 using amylase solution prepared at pH 6, then 7, then 8. Question 3.2, what's the independent variable in the investigation? We should know that independent is what we change in the experiment, and we've said it quite clearly here. What are we changing? We're changing the pH of the amylase solution, or you could say pH of the you could actually just get away with just saying pH, but yes, pH of the amylase solution. Describe how the student would know when all the starch had been digested. So this is knowing about our food test. How do we test for starch? Well, we need to know we would use iodine. So we would add iodine, and to get for 1 mark you have to say the colour change you would see. Well, if all the starch has been digested, then the iodine would actually stay orange. So you would add iodine and it would stay orange, or you could say it would not turn blue or black. Because remember, if starch is present, it changes iodine from an orange colour to a blue-black colour. Now that is if it's positive, but if there is no starch present, as in this situation, it would stay orange. So when all of it's been digested, it stays orange. Question 3.4 then. The figure 6 shows the student's results. So you've got time taken to digest all of the starch at your different pHs. It then says what is the optimum pH for amylase, okay, using figure 6. Now in this graph, if we're working out the optimum pH, we're working out the best pH. The best pH would be the time that it does it the quickest. A lot of people, when this paper came out, went to this point here. But actually, this would be the worst performing pH because it's taking the longest amount of time, 10 minutes to break it down. The optimum pH would be the shortest amount of time, which would be this point here. You read down on your graph and you've got to say it accurately because they've given you the scale, which is quite easy to read. It would be 6.8. If you said 6 or 7, you wouldn't get the mark. You've got to say it exactly from this graph. A student then did a different investigation. This is the method they used. They prepared amylase solution at the optimum pH. They mixed the amylase solution with starch in a boiling tube. They measure the concentration of sugar every 10 seconds for 2 minutes. Figure 7 then shows the student's results. So you've got the time in seconds and you've got the concentration of sugar in arbitrary units. It says, how much time did it take for the amylase to digest all of the starch? Okay, so we know that starch gets broken down into glucose or sugar. So at what time did it take to break down all of the starch? Well, the starch stops going up at this point here. So I would be saying that the starch is when it goes totally flat. Actually, it's probably a little bit after 80 because it's still slightly going up at 80. And I would say it's definitely gone flat at this point here, which is 82 seconds or 84 seconds. You're actually looking at the mark scheme, you're allowed any time between 80 and 84. Okay? So as long as you put an answer between 80 and 84, you would then get a mark. Question 3.6, determine the rate of sugar production per minute at 40 seconds. Okay, so the rate of sugar production at 40 seconds. Now it's asking you to find the rate of a reaction at one specific moment in time. Now if I go to 40 seconds, I've got a curved line here where it's asking me to find the rate. Remember, rate on a graph, when you've got time in it, is basically equal to the gradient. But you can't find the gradient of a curved line very easily. What you then have to do, or remember, you've got to draw a tangent on. You get 1 mark for recognising that if you draw a tangent at 40, okay, you would then pick up 1 mark because you recognise you need a tangent to find the rate of a curved line. What you then do is you take your tangent and then turn it into a triangle. Now it actually doesn't matter what angle you draw your tangent at. You get 1 mark for drawing the tangent and then 1 mark for using whatever size tangent or whatever slight angle you've done it at. Even if you do it at a slightly weird angle, you'd still get plenty of marks through doing it. So what you then have to do is calculate your change in your y value and your x value. So obviously you would do this much more accurately using a ruler, but I can see I'm going from 2.6 up to 4.55. So what I would then do is calculate the change in my y value. So 4.55 minus 2.6 gives me a change of 1.95. On my x-axis, I've gone from 20 seconds and I've drawn it to about 60, probably about 60. Obviously you guys would do it with a ruler, be much more accurate. I've probably got to about 63. So I've gone 20 to 63, so therefore my change is 43. So therefore, to calculate the gradient of a line, this part is still the same as if it was a straight line. It's your change in your y value divided by the change in your x. So my change of my y value was 1.95. My x value was 43 seconds. So 1.95 divided by 43 gives me a change or a rate of sugar production of 0.045348884. Okay, now that is how much sugar is made per second. Okay, so if I take that down to my answer area, I know per second I'm making 0.045348884 per second worth of sugar, because my graph is in seconds, but I want the rate of sugar production per minute. So what I would simply do is recognise 60 seconds in a minute times whatever answer you get, and obviously everyone would get slightly different answers and different numbers. That is fine. It's more on a tangent question, they're more interested about the actual method rather than necessarily the exact accuracy of your tangent line. So times it by 60 gives you 2.7209 grams per arbitrary units per minute. Now it hasn't said anything about decimal places, so I might just leave it as 2.72. Okay, and that would be my final answer. Now odds are when you do this yourself and try this question, you might get totally different random answers that are slightly different from 2.72. Don't worry about that. You get 1 mark for working out or recognising and drawing a tangent. You get 1 mark for then getting your values and recognising it's y over x. You get 1 mark for getting to your answer of whatever, as long as you've pulled off your values correctly and getting to your final answer per second. And then your final mark out of the 4 is getting it per minute and recognising you times it by 60. Question 3.7 then, a 6 marker. Explain how the structure of enzyme molecules is related to the effect of the pH on the activity of amylase. So explain how the structure of the enzyme molecule is related to the pH on the activity of amylase. So this is basically saying to me, describe the structure of enzyme molecules and how it is affected by pH. So I'd be thinking of basically all of my knowledge to do with enzymes. So I should recall that enzymes in a normal, if it's in optimum conditions, enzymes are proteins, okay, and have a 3D structure. And then I'd be thinking, I could draw out a little diagram to myself, okay, remembering enzymes a bit like Pac-Man. Enzymes have a 3D structure, and it's a bit like, act like a lock and key. So enzymes act like a lock and key with the substrate. Enzymes have an active site, so the part of the enzyme where the substrate fits in is called the active site. And remember, substrate is just the food or the component or the molecule that goes into the active site. So you've got the active site, and then you've got the substrate. So enzymes have an active site where the substrate enters. Okay, and they can fit together. The substrate can go in because they are the same shape. They have got complementary shapes. So enzymes can bind with substrates if they have complementary shapes. Okay, and this is what then binds with it. It would then break it down. So I've described, first of all, how the structure of the enzyme and how it works normally. Then I need to consider the effect of pH. So you're thinking, what happens to enzymes if they're put into pHs that are slightly different? If enzymes are put into pHs which are different from the optimum pH, they can, and the key word you need to say is denature. So remember, denaturing is when your enzyme basically loses its shape, it breaks down. It doesn't die because they're not alive, but it denatures. This causes the shape of the active site to change. Because it's now changed, our substrate cannot fit inside. The substrate cannot bind or fit inside. They no longer have complementary shapes. So if they no longer have complementary shapes, what is then going to happen to the effect of the activity on the amylase enzyme? Well, if they can't bind, it's going to decrease the rate. This decreases the rate of amylase or enzyme activity. So, in this question then, explain how the structure of an enzyme molecule is related to the effect of the pH on the activity of amylase. Start off by describing your standard lock and key enzyme model and how it would normally work, and then break it down onto the effect of the pH You know that it denatures. It's the same principle with changing the temperature. If it gets too hot, it also denatures. Okay, so that is how I would tackle that 6 marker. Question number 4 then. Photosynthesis is an important chemical reaction in plants. Why is light needed for photosynthesis? Well, we should remember that light is a source of energy. So you just say light is for energy needed for the reaction. And as long as you put that and get across that it's light is the source of energy, that's enough to get you the mark. You could also say photosynthesis is an endothermic reaction, so therefore it's absorbing heat from the surroundings. That would also be accepted. But I think the key thing is recognising that light is the energy source. Question 4.2, what is the equation for photosynthesis? Well, we should remember that plants take in carbon dioxide from the air, they react it with water, and they produce glucose, which is the food, the whole point of photosynthesis, and oxygen. So I just look for those symbols. Carbon dioxide, okay, and water, okay, makes glucose and oxygen. So glucose and oxygen. And the only one that has them all in the right places would be the third one down. Remember, you balance it just by putting the 6's in front of the 3 smaller molecules. Okay, question 4.3 then. So a student investigated the effect of different colours of light on the rate of photosynthesis at room temperature. The student used pondweed in water. A piece of pondweed was placed in red light, then blue light, and then green light. Each colour of light was the same light intensity. Describe how the student should make accurate measurements to obtain valid results for the rate of photosynthesis. Okay, so in this practical, you should be thinking of your required practical when you're measuring the rate of photosynthesis with pondweed. Now, the basic method, which is not accurate or not valid, is using when you've got a pondweed in water. Okay, so if you pondweed set up within the water, and then you count the oxygen bubbles coming off. This, counting bubbles, is not accurate. So counting bubbles is not accurate. If you started describing a method of counting oxygen bubbles, you would automatically get 0 marks, because that's not the accurate way of doing it. What you should be thinking of is a slightly different method where you get your beaker and then you get a funnel, put that at the bottom of your beaker, and then you have either attached to your funnel a inverted measuring cylinder which is submerged in water. So you can have a measuring cylinder over your funnel, and then as the photosynthesis occurs and all the oxygen bubbles are released, it pushes the water down out of the measuring cylinder, and you can record the amount on the scale. The other way of doing it is not to bother with a measuring cylinder, but just simply attach it to a gas syringe. So you would just have connected to your funnel a gas syringe, which can then be used to simply measure how much gas you've got being produced. And as the oxygen bubbles from your pondweed are being made, it pushes back your gas syringe and you can just record that scale. So you need to be thinking about that method because it's more accurate and more valid. Because obviously the bad things about the bubbles experiment is that you can miscount bubbles, and bubbles are not always the same size. So what I would be saying is, you would say, set up pondweed with a funnel submerged in water. Okay, so immediately you've told the examiner you are working on the accurate method. Okay, and then I would say, keep it simple, connected to a gas syringe. Now, you may not have done it this way in class with a gas syringe, but there's nothing wrong with saying it, and it's the most accurate way of doing it. So what I would then do, after I've set it up, is allow the pondweed to equilibrate to the surroundings. Basically means get used to the surroundings that it's in, or equilibrate to the light intensity. Get it to a stage where it is consistently photosynthesizing. What you would then do, after you've got it into a certain conditions, you can then allow the pondweed to photosynthesize for a period of time. You can make it up, it's your method. I might say an hour, and I would say if you're saying leave it for an hour, using a clock or a stopwatch. Okay, so use a stopwatch to measure the time. You would then measure the volume of gas in the gas syringe. So you have made it accurate because you're using a gas syringe, 1 mark. You've allowed it to equilibrate to the light intensity. You haven't just put the pondweed in and just let it go. You've allowed it to settle to the conditions, and you've recorded the amount of oxygen made in a certain amount of time. That has all made it accurate. Now, to make it valid, you need to be making sure that you are, what we would normally do in an experiment, repeat it 3 times. So you would change the light intensity or change the light colour. And then you would repeat 3 times. And then after you do your repeats, you calculate an average or mean. And you can also say you would remove anomalies. And that would be your last mark. Okay, and through doing that, that's increasing the accuracy because you're controlling the control variables and you're increasing the validity of it. A scientist investigated the effect of different wavelengths of light on the rate of photosynthesis, and you're told the wavelength of light determines the colour of the light. So figure 8 shows a student's results, figure 9 shows a scientist's results. So if you look at the student's results first, you can see the colour of light and the rate of photosynthesis in arbitrary units. So you've got red and blue, pretty similar, but green light, okay, not much photosynthesis at all. If you look at the colours of or the scientist results, you've got the wavelength of light and you've got the photosynthesis in arbitrary units, and you can see how it changes with wavelength. It then says, why are the results of the 2 investigations presented differently? So we've obviously got a bar chart and then we've got a line graph. So why have they been presented in this way? Well, this is just a fact that you need to know, that if you've got categoric data, so categoric data is when you basically, your independent variable is words or categories. Okay, that means you would present it as a bar chart. Okay, so your independent variable, if it's categoric data, so like names of football teams or favourite colours, or in this investigation, the colour of light shining on the pondweed, because it's not numbers, they are words or categories, categoric data, you would draw a bar chart. But on this one, we're measuring wavelength of light. So the wavelength of light is a number. This means our independent variable this time is continuous data. So continuous data is when you've got numbers as your independent variable. Okay, I'm changing the number, the wavelength of the light, and I'm measuring the rate of photosynthesis, and that is why I would then draw a line graph. So if I had to summarise this into an answer, I would say the student's investigation has categorical data as the independent variable, and therefore has used a bar chart, whereas the scientist's results has continuous data. And therefore uses a line graph. It then says, suggest the range in wavelength of green light. Use figure 8 and figure 9. So I've got to kind of estimate what I think my wavelength of green light is going to be. So if I go back to the graph, I can see that green light has very limited rate of photosynthesis. So I go to my second graph, and I would say where you've got to, it gives you quite a large range on this, is basically where there is low amounts of photosynthesis happening. And they actually give you a very large range you can say on this. You can say any number between 475, which is this point here, for where it starts, to 525, which would be this point here. So any number between these parts would be where it starts, and then when it finishes is 575, okay, to 650. So that is quite a, which is this point here. So that's quite a large range you've got for both values. So it says it's exactly 492 to 577 on the mark scheme, but really you're allowed any number between 475 to 525, and then 575 to 650. So I'm assuming when people did this paper, there's an awful lot of different answers, and therefore they reflected that in the mark scheme. Okay, so that's the end of that. Okay, so question number 5 then. This question is about tumours. Describe the similarities and the differences between benign tumours and malignant tumours. So 4 marks here, similarities and differences. Therefore, I can assume there's 2 marks discussing the similarities, 2 marks by talking about the differences. So I would give it a little subheading and say similarities. What do I know of the similarities between these 2 different types of tumours? Well, you can think about how tumours are made, okay, or what are the causes of them. Tumours are formed by mutations in the DNA, okay? Tumours are formed or caused by mutations in the DNA. That is the same between benign and malignant, so therefore that's a similarity. You can then say what an actual tumour is, because it doesn't matter whether it's benign or malignant. What is an actual tumour? Tumours are both caused by cells that are dividing out of control, forming a lump of cells. Okay, a ball or a lump of cells. Okay, and that is where I would leave it. Those are your 2 similarities. You've said what they've been caused by, and you've said that what a tumour is. So then we need the differences between benign and malignant. Now, hopefully you remember that although both are awful, if ever you were going to get a tumour, you'd want to be getting a benign tumour, because benign tumours are not cancerous. So I would say benign tumours are not cancerous whereas malignant tumours are. So that's one difference between it. What this actually means is benign tumours do not spread around the body, and make secondary tumours, but malignant tumours do. And that is what makes them cancerous. Okay, so that is two differences. That would be enough to get you 4 marks here. Okay, one other thing you could say is that benign tumours are closer to normal cells than malignant. But I would leave it as these 4 points here, and that would get you 4 marks there. 2 similarities, 2 differences. Figure 10 then shows data about skin cancer in the males and females for different age groups in the UK. It shows the number of new cases of skin cancer in 1 year and the number of people with skin cancer per 100,000 population in 1 year. So you've got the graph, and it's giving you a lot of information here. You've got age in years, and there's 2 axes. You've got the number of new cases of skin cancer for males and females, which are your bars, and then you've got some lines, which are the number of people with skin cancer, okay, per 100,000 of the population. So what I would be doing is breaking it down and seeing what part of the graph are actually telling you what thing. So you can see on your first y-axis, it's about the new cases, which I've highlighted in yellow. The right side of the graph is the number of people with skin cancer per 100,000 people in the population, and that's what I've highlighted in green. So the lines are representing or linked to the green part of the graph, the right y-axis, but the yellow part is related to the left. So take the time, make sure you understand this graph before you then dive into the questions. It then says there are no new cases of skin cancer diagnosed in people younger than 15 years of age. Explain why. Okay, well, if we go back to this graph then, we can see the number of new cases. So I'm looking at my y-axis. Below 15, there's no bars, there's no cases of skin cancer. So it's saying suggest why very young people tend not to develop skin cancer. Well, think about what skin cancer is caused by. Skin cancer is caused by UV radiation, okay, or ionizing radiation hitting a person. So therefore you might say a young person has limited exposure to ultraviolet radiation or ionizing radiation. You just need one of them, really. So they haven't been exposed to much radiation. And therefore, because it's a 2 marker, so you've made your point, then you need to explain it and think about, right, what does ionizing radiation do to the DNA? Therefore, there is less damage to the DNA and fewer mutations. And that would get you 2 marks there. So you made your point and then you've explained it. Give 2 conclusions about the number of new cases of skin cancer using figure 10. So in this question, I'm just looking at the new cases of skin cancer in figure 10. And this is where people lost the marks in this paper when they used the wrong part of the graph. So I am purely just looking at the bit I've highlighted in yellow, which is my bars, and I'm looking for the pattern in this graph, which is just referring to the bar charts, okay? 2 conclusions about the new cases of skin cancer. So ignore the lines, the dashed line and the solid line. You've got to kind of imagine that they're not there, because that's not relating to this question. You're just talking about the new cases of skin cancer. So I can see, as my bars go up, I can see, right, my white bars are higher, generally, up until 55 to 59, and then, when it gets to 60, it's actually then lower. So the white bars, which are for females, are always higher up until 59, 55 to 59. So that would be my first conclusion. I'm just going to write the answer here, but this is 5.3. Okay, so 5.3, I would say females have more cases or more new cases of skin cancer between 15 to 59. Okay, then I can see after 59, actually all of the grey bars are bigger. Okay, which means that the males are then having more cases of skin cancer. So that could be my second conclusion. After 59, okay, so going from 60 to 64 onwards, males have more new cases of skin cancer. Okay, so you've looked at the correct part of the graph, you've identified the key part that you actually need to describe, and you've pulled off two conclusions. Now that would be enough to get you the 2 marks on the 2 points here. You could also say, as a third point, okay, just actually say which age group has the most new cases of skin cancer. So for a different point, you could have also said that ages 65 to 69 has the most new cases of skin cancer. Okay, so this was a hard question because a lot of people started talking about the dash line and the flat line, about the cases per 100,000, but that's not this question. So we can say we've done that one. It then says question 5.4. The data for the number of people with skin cancer is given per 100,000 of the population. Suggest why the data is not given as the total number of people. Well, what you've got to imagine is, in the country, that is the population of each of these different age groups exactly the same? No, there's generally more old people than there is young people, because we're living longer. So therefore, what we would say is, just simply, each age group has different numbers of people, and therefore that would, if we're doing it per 100,000, would account for the different group sizes. Okay, so therefore doing it 100 per 100,000 people makes it more comparable. Okay, if you had different group sizes, it would massively screw your graph. So then you've got figure 10 repeated below. Okay, so figure 10 is then repeated below. You've got the same graph, same information, they just give you a fresh copy of it in case you've annotated the previous one for the previous question. It then says, describe 2 trends shown in the data in figure 10. But I'm only using the data for the number of people with skin cancer per 100,000 of the population. So I can only refer to, this time, my dash line and my solid line. I'm ignoring my bars. So again, I'm looking for 2 trends. Well, the trend, I would say the obvious thing, that is in front of you. What's happening to both lines? They're increasing. The overall trend or pattern is, as age increases, the number of people with skin cancer per 100,000 increases. And that is the case of in males and females. So that would be my first point. Always say the obvious mark, okay? And then you've just got to find one more. What I could then say is, if I'm looking at my female line, okay, The female line, the number of cases of females with skin cancer, is higher than males up until the age of 59. So my female line is higher up until the age of 55 to 59. Then the males then accelerate and take over the females. So that would be my second point. The number of females with skin cancer is greater from 15 to 55 to 59, okay, than males. But after 59, the amount of males increases steeply. Okay, so it's then accelerating and then becoming much higher. So that would be my second mark there. Question 5.6 then. The estimated population of males age 80 to 84 was 694,000. Calculate, the estimated population of males age 80 to 84 was 694,000. Calculate the number of males age 80 to 84 with skin cancer in that year. Using figure 10, give your answer to 3 significant figures. Okay, so I know the total amount of people, and I want to estimate the number of people with skin cancer in that year. So I'm looking at my 80 to 84 category for males. So what I would then do is go back to my graph. I might need to just get rid of my annotations here quickly. So I know I'm looking at my male line, which is my dash line, and I'm looking at my 80 to 84 category, and I'm looking therefore at this point here. So what I would then have to do is read across, and remember I'm using my axes on my right hand side, and then read it across. So that is then telling me, if I read that accurately, it's 5 little squares divided by 20. 20 over 5 is 4, so it would be 1 little square below 120. 120 minus 4 is 116. So therefore I know on average there is 116 people per 100,000 of the population that is then got skin cancer. But if I know the total amount, what I could then do is 116 times by the total population, 694,000. But remember, it's per 100,000 people, so divide it by 100,000. And that would give me an answer of 805.04, and therefore giving it to 3 significant figures. Well, you draw your line and you know, well, 0, so it's just 805. And that is your final answer there. Okay, so question 6 then. This question is about the heart. Why is the heart described as an organ? So this is your definition of what is an organ. You need to remember that it goes cells, tissues, organs, and then you've got organ systems. So what is an organ? Well, it's a group of tissues working together. So you would just say an organ is a group of tissues working together to perform a job or function. Now you need to say both parts to get the single mark here. Group of tissues working together to perform a function. Valves in the heart keep the blood flowing in one direction. Figure 11 shows the heart with one of the valves labelled. Explain the effects on a person if a valve labelled in figure 11 develops a leak. So remember, valves stop, as it's told you here, keeps the blood going in one direction. So if you've got a leaky valve, then it might cause the valve of the blood to go in the other direction, which would be your first point. The valves, or the leaky valve, may cause blood to flow backwards. Okay, now if we remember that this is our left side of the heart, so it's got all the oxygenated blood going round it, you've then got oxygenated blood flowing backwards into the heart. What does that then mean? Well, it means you've got less oxygen flowing around your body. So what I would then say is that because you've got the black flow of blood, okay, less oxygenated blood is then transferred around the body. Because we therefore have less oxygen, what is that oxygen then used for? Well, it's used for respiration. This means less respiration would occur, and therefore if you have less respiration, therefore you have less energy. Therefore, because we've got to bring it back to the person, therefore this person would have less energy and be more tired. Okay, now that would be enough to get you 4 marks there. Okay, so last question on this paper then. Faulty heart valves can be replaced using biological or mechanical valves. The faulty valve is replaced during an operation. It tells you biological valves are from animals or human donors and they allow blood to flow through them normally. They may wear out and stiffen over time, so may need to be replaced. Mechanical valves are made from synthetic materials. They may cause blood clots on the surface of a valve. They require anti-clotting drugs to be taken for the rest of the patient's life and can last a very long time in ideal conditions. A woman enjoys extreme sports and would like to start a family. The woman needs a heart valve replacing. Describe the advantages and disadvantages of this young woman having a biological heart valve instead of a mechanical heart valve. Okay, so what we need to do is make sure we are giving biological heart valve advantages and disadvantages, but also referencing and comparing it to a mechanical heart valve. So what I would potentially do is almost draw it out as and just do it as a little summary list of every reason you can think of. So I would say positives of biological heart valves. Now I'm just going to do it as bullet points, but obviously you turn it into short sentences. So advantages of biological is that it allows blood to flow through them normally, whereas mechanical could cause blood clots. So I would say a clear positive would be with biological has no risk of blood clots. Okay, so that's one clear advantage, and you could compare it to linking mechanical, whereas mechanical does. A disadvantage of biological, okay, so it's 4 marks. So a disadvantage of biological is that they might need to be replaced, but mechanical can last for a very long time. So a negative is that for biological valves may have to be replaced, whereas the mechanical one would last a long time. Okay, then I would look at some of the disadvantages of mechanical heart valves, where you could say it requires anti-clotting drugs to be taken for the rest of the patient's life. So therefore another advantage of biological is that there would be no drugs or needed for the rest of the life, no anti-clotting drugs needed for the rest of the life, but you would do with a mechanical heart valve. And therefore, linking it back to extreme sports, okay, well, if you're taking anti-clotting drugs, then that could be bad if you want to continue that, because if you get an injury, you would then be bleeding an awful lot. Okay, so I would be saying a disadvantage of mechanical, and therefore an advantage of biological, is that if injury occurs, there would be, and you're on anti-clotting drugs, a lot of blood loss, which could make it more dangerous. Okay, so you just basically need to pull out some advantages from the information that they've given you there. Again, question, end of the paper, gives you loads of information, and you just need to try and structure it into a real clear, coherent answer. So that then brings us to the end of the questions. So that's the end of Biology Paper 1, higher tier from 2022. Hope it was useful. Thank you very much.